Okay, hello everyone. Today I will be talking about some of the research I got to conduct this summer in the lab of Dr. Sheila Singh, who is a pediatric neurosurgeon scientist at McMaster University in the Center for Discovery in Cancer Research. And so the project that I undertook this summer was titled The Role of uPAR in Standard of Care Resistance and Recurrence in Glioblastoma. So GBM stands for glioblastoma, and it's WHO grade 4 glioma. Um, Originally, when patients present with a tumor via MRI, they undergo surgical resection, at which point pathology diagnoses it as GBM. And so the standard of care for glioblastoma, when they find out and diagnose that it is in fact a GBM, is they receive the surgical resection, followed by chemoradiotherapy. The chemotherapy that is used is temozolomide, and then they also use a combination of radiotherapy. And so usually the tumor begins to shrink after this time, but unfortunately, in almost all cases, recurrence of the tumor occurs, and there is no established standard of care at recurrence. And so, unfortunately, this devastating disease has a median survival of 8 months. And so there needs to be advances in possible treatment options at recurrence due to the very low average survival of this disease. So a lot of the work that the Singh Lab does at McMaster is founded on the work that Dr. Singh did during her PhD in the lab of Dr. Peter Dirks. And so they identified a subpopulation of GBM cells called brain tumor initiating cells, and these are known to seed the tumor. And so they identified these brain tumor initiating cells by the presence of a cell surface marker known as CD133. And this is quite important as for our work. And so the reason this is important is we believe that brain tumor initiating cells are the root of the problem. And so what we believe is GBM is extensively heterogeneous. There is intratumoral heterogeneity, so within the same tumor, and there's intertumoral heterogeneity, so across tumors. And that exists even between the primary and the recurrent GBM. And so this kind of schematic represents like the primary disease, where standard of care is initiated. So after diagnosis of GBM, followed by the recurrent disease. And so if you were to look at this schematic, what would start as a purple cell kind of changes its phenotype, and then there's some expression of some blue cells, some green cells, but in addition, there's also these pink cells and orange cells and dark blue cells. And so even at the primary disease, there's so many different what we call clones that exist, and these have different expression of cell surface markers, and is part of the reason why it's so hard to provide one therapy to treat the disease. And so after treatment initiation, we believe that these brain tumor initiating cells, some of them are eradicated. So you can see the light blue, dark blue, and green have been eradicated. But there is this subpopulation of these brain tumor initiating cells that are resistant to chemoradiotherapy. And in fact, continue to kind of expand their own like clonal basis. And so our lab focuses specifically on culturing these brain tumor initiating cells. So we will get patient tissue from the hospital in collaboration with the neurosurgeons and neuropathologists there. And we will culture these using stem-like conditions to try to select for these stem-like populations of cells. And so what's quite interesting is in our lab, we have tons of samples, and a lot of them get made into cell lines, which can be used for different profiling experiments, different sequencing. And so the pictures that I've shown on the slide actually represent one pair. So BT594 and BT972 are the primary and the recurrent disease, respectively, of a single patient. And so if you look within the dish, like a very zoomed in scope of one dish, you could see that within the dish, not all of these neurospheres look the same. And so these brain tumor initiating cells are known to proliferate and self-renew and propagate as neural spheres in most cases. But you could see that within even one tumor cell line, not all the cells look the same. And then when you look at its recurrent, it looks completely phenotypically different. And so this kind of proves, if you look within the same tumor, like even though these are the same patient, at primary disease, there is a lot of intratumoral heterogeneity, but even looking between two tumor samples, primary and the recurrent, there is intertumoral heterogeneity, and this is why there needs to be therapeutic advances for GBM that kind of look at this heterogeneity and try to create a solution to the problem that considers this. So through whole cell proteomics, done in collaboration with the lab of Dr. Thomas Kisslinger, using the cell lines that I showed on the past slide, what we saw was when you compare the proteomic profile of BT972 and BT594, there's this gene which codes for a protein called uPAR, which is highly expressed in the recurrent disease. And it's highly differentially expressed. Whereas that is in comparison to the primary. And so what we went to show was using these, using our cell lines, so MBT173, I don't know if you can see this, but MBT289 and BT972, we performed flow cytometry, which looks at cell surface expression of proteins. And what we did was we profiled for the cell surface expression of uPAR. And so what we see is there's a high percentage or high proportion of the cells that exist within the dish that are uPAR positive in the recurrent GBM. And so this so originally we had looked at specifically BT972 because we saw differential expression, and we started to see a trend with looking at all of the recurrent GBM samples we have in the lab, and we see that it's consistently highly expressed in the recurrent disease, making it a possible immunotherapeutic target, but also something that's quite important to try to understand the deeper biology between why it exists so highly at recurrence. So what we did in the lab was we posed the question, does uPAR play a role in standard of care resistance and recurrence in glioblastoma? And this question was really prompted by that whole cell proteomic analysis of the same patient from BT594 to BT972, just from the primary disease to the recurrent disease. Why is there a differential expression of this this protein called uPAR? And so what we had done is we conducted an in vitro standard of care protocol. So this only consisted of the chemoradiotherapy portion of the standard of care for glioblastoma, but this is what we could best recapitulate in the lab. And so what we did is we plated primary GBM BTICs, which are treatment naive. So as I had explained prior, the surgical resection often occurs after presentation on an MRI. So the patient has not received any temozolomide or radiation because the diagnosis usually occurs after the actual resection of the tumor. And so after plating these GBM BTICs, we added the temozolomide at a very particular concentration and left in the incubator for 1 hour. And then we used our irradiator to irradiate the cells with 1 gray of radiation. And so this procedure was repeated for 4 consecutive days, totaling to 5 days. And after this, we allowed the cells to recover for 3 additional days. And so after this whole set of experiments, what we did was we either analyzed cell surface uPAR expression by flow cytometry, comparing it to a control primary GBM BTIC that had not underwent standard of care treatment and only got the control treatment, or we read out the plate using Presto Blue, comparing proliferative capacity of the cells. And so this was slightly modified from a protocol that existed within our lab. So the very first thing we went and did was we validated that this in vitro standard of care protocol actually worked. And so what we see is in 3 primary GBM tick lines that there is significantly reduced proliferative capacity of the standard of care treated GBM ticks as compared to the control group. So we know that the standard of care was doing what we believed it would do, as there was reduced proliferative capacity. And so then we could move forward, trying to figure out if cell surface uPAR expression increased after standard of care. And so after performing the treatment on the cells and letting them recover, what we see is in MBT06 and MBT433. So in MBT06, you see a trend towards an increase in uPAR expression. And so this is demonstrated by the red histogram plot. And a similar thing in MBT433, you see a significant difference in uPAR expression in the standard of care treated arm of this experiment. So we know that it appears as if uPAR expression is increasing after standard of care. And so this was consistent with what we originally saw with BT594, BT972 differential proteomics. So the next step for this project, and so what I will be conducting next summer is looking at if you knock out uPAR using CRISPR-Cas9 in our GBM tick lines, if standard of care becomes more effective. So if actually affecting the expression of uPAR is important to the population of cells that survive standard of care. Then we're going to perform in vivo survival studies using the CRISPR-Cas9 knockout of uPAR in recurrent GBM tick lines, so where we see it's highly expressed, and we're going to inject it into a patient-derived xenograft model to try to see if the mice, which is the model we use NSG mice, which are immunocompromised mice, whether there is a significant survival advantage of mice that don't have this uPAR protein. And finally, we're going a very exciting angle of this project is we're going to validate uPAR as an immunotherapeutic target using anti-uPAR chimeric antigen receptor T cell therapy. And so that's CAR T cell therapy, which is a popular form of immunotherapy and is becoming more widely used. We're going to try this in vivo using the recurrent GBM tick lines injected into a patient-derived xenograft model. And all of this work is done with the great help of everyone in the lab. And in our lab, we have 3 teams, which are Team Glioblastoma, which is what I'm a part of, Team Medulloblastoma, and Team Brain Metastasis. And everything was, there was great help done by my supervisor, Dr. Sheila Singh, and the senior research associate in the lab, Dr. Chitra Vanugopal, who really helped me troubleshoot any issues that occurred during my time conducting this project. And we would also like to thank all of our partners that really support the research we do, of which Brain Tumor Foundation of Canada is quite important. And this work would not be possible without all the help that we get. Thank you. And if anybody has any questions, please contact Susan Ripers at Brain Tumor Foundation of Canada.